Okay, thanks, Sonia. Thanks very much for having me. It's a pleasure to be here. I hadn't heard that Bob Konopaz was coming yesterday, so hopefully we're not going to be too redundant. He probably took all the good stuff for himself, and we didn't review the presentation. What I'm hoping to do today is to share some of my personal perspective on the importance of technology development to biotech manufacturing. And I hope you come away with the view that process technology is going to continue to play a very important role in biotech's future. In particular, in light of the business and, for want of a better word, political drivers, political meaning healthcare system-oriented, cost-oriented drivers, I believe it's going to be very important. I'm going to show you both try to make a case for the importance of technology as we look at it at Wyeth, and also to provide you with some data and some of the areas of focus of my lab in cell and molecular sciences in trying to address these important challenges. So, by way of bio, maybe the history of Wyeth and my own sort of relate. I did a PhD in molecular biology from the University of Virginia and a postdoc at Monsanto, actually studying developmental biology of limb regeneration in the red-spotted newt, but from a molecular biology perspective, and joined Genetics Institute in 1990. Genetics Institute is one of the oldest biotech companies, was founded in 1980, and we worked on all of the recombinant proteins of the day, Factor VIII, TPA, EPO, et cetera, in those days. The company was acquired by what was American Home Products at the time, beginning in 1991, after we actually lost a major patent battle with Amgen on erythropoietin. And that basically was close to licensure at the time, and at the final Court of Appeals for the Federal Circuit, we lost that battle. We needed money. American Home Products purchased 60% of the company at that time, and then completed the acquisition of GI, something like 96, I believe it was. So we became basically what had been an early biotech company and got incorporated into a large pharmaceutical company, which is now called Wyeth. Another interesting fact is that in the mid-90s, American Home Products also held a majority stake in Immunex, which later was purchased by Amgen, and this is where Wyeth got a stake in Enbrel, which is a large rheumatoid arthritis drug, just that people may or may not be aware of. So we formed a unit called Wyeth Biopharma, basically to bring development and manufacturing together because we think the technology is so specialized and have made major investments there. So I'm speaking to you today, and my perspectives on technology are coming not from a small biotech, but as someone that's in a large biotech firm within a pharmaceutical company, which is sort of important because there are different drivers in different places in the industry. And that's just something to keep in mind. We do have a number of commercial products. Enbrel, I mentioned, is the huge one, but a number of other commercial products, including several coagulation factors, that have powered us to a position of over $3 billion in revenue, which many people don't know with the name Wyeth, but as a standalone company, we'd be the fourth largest biotech company in the world. This slide is just sort of illustrative. We all know that the basic steps, and you might call this a now traditional biopharmaceutical process, where we take genes, put them together with cells, grow the cells. The product is these little triangles secreted from the cells, and then the protein has to be purified. Usually we use chromatographic steps. I'm sure you've seen all of this before. Purify that, then put it in a vial to deliver it to the patients. I think the important thing to remember is that, to remind ourselves, is that in biotech, the really unique thing about biotech in the traditional sense of the word that separates it from traditional pharmaceuticals is the fact that cells actually synthesize the product. And that fact really has been an overarching and an impactful fact on the industry all the way along. The processes, therefore, that are required in order to do this are very complex, and the facilities, as I'm sure you know, are also very complex and expensive. And from my point of view, one of the major problems with pharmaceutical biotech over the years has been what some people would call the capacity problem. The lead time for building facilities and the cost of building facilities is so extraordinary that it's really a huge barrier to entry for everybody, and a huge financial risk. And it drives the financial guys crazy because you can never start building. If you start building early before you know that you need the capacity, you could end up with a huge amount of unused capacity. Conversely, if you wait too long, you could end up with a huge amount of product being unsold, which actually was a problem with Enbrel in the early days because there wasn't enough capacity to meet demand, which was costly and drove everybody crazy. It's still a very successful product. So that's really an overarching issue for us and one that drives our interest in technology. The other point just to make is I'm a molecular and cell biologist by training, so I'm an upstream guy. I've been working in process technology since 1990, primarily on the upstream side, so my remarks are really oriented toward that side. I think Bob may have mentioned some of the challenges. The better we do on the upstream side, the more we're putting pressure on the downstream side to keep up. And that's something that we're definitely going to have to deal with. We use cells to make product. There are many different choices of types of host cells. I'm sure you've seen all of this. Basically, all of these work very well. From our perspective at Wyeth, we're really heavily invested in animal cells, CHO cells, and we expect that this will remain our predominant platform for at least the next 10 to 20 years. One of the reasons for that is, if you remember, any product that we begin to develop today will not get licensed for at least 7 years or longer. And so, even if you give it a decent product life cycle, that's the kind of time frame we're looking at. And we are starting our product development now with CHO cells, basically almost exclusively, largely because of the stable of molecules that we're developing. All of these options are useful and probably of value in different circumstances. We did look at the possibility of transgenic goats and actually did a project where we compared the expression of a monoclonal antibody in CHO cells and transgenic goats. We got excellent yield in our CHO cell bioreactors, in this case 3.4 grams per liter, and nice yield in the milk of transgenic goats, 5 grams per liter. But for us, what this translated into is if we go the traditional route, our Grange Castle facility, I'll show you a picture if you haven't seen one coming up in a slide or two, that has 6 12,500 liter bioreactors. In a year with this product, we could make 4 tons of material, which is an incredible amount. In transgenic goats, it would take a herd of 6,000 goats, if I've got the math right here, to do that. And for us, with the infrastructure and investment that we've already made, this option is much more attractive based on return on existing investment and risk relative to transgenic goats. For us, even though this technology you may have heard, I think there's a licensed product now in Europe, or it's close to licensure in Europe from Genzyme Transgenics. This technology is moving forward and does have merit, in my opinion. It doesn't make sense for us right now. So we're going to be focusing on CHO cell technology as our primary area of interest, while we will continue to survey, and my lab is responsible for continuing to keep an eye on developments in all of these areas and trying to see where there might be a niche where they fit in. Yes? We have used insect cell lines for early research and preclinical production, but we haven't, in terms of taken it seriously in terms of looking at it as a manufacturing option for clinical production. We're not alone in this. The majority of biotech products are made in animal cells, although there's a good distribution. And actually, the next point that sort of drives our thinking toward technology, begins on this next series of slides and start to look at the market a little bit. This illustrates from one point of view that this is a huge market. These 5 of the top 20 products, pharmaceutical products, are biotech products. They number in the billions of dollars. These are all made in CHO cells, which sort of illustrates the importance of CHO cell technology. It's also worth looking at the fact that biotech is growing faster than the traditional pharmaceutical market. So as you see, a couple of these classes of drugs are growing much faster. And if you look at the pipelines of the biotech industry as a whole, there's a lot coming forward. Many of these products are antibodies and fusion proteins that are going to be made in mammalian cells, and so there's going to be additional pressure being put on the system by these products. And another factor is that many of these products are administered chronically, which means more doses per patient per year, which means the need for more protein. And essentially, this is putting a lot of pressure on the industry and will put a lot of pressure on the healthcare system as a whole, which is a big concern of ours at Wyeth. And for me personally, we think that these medicines are very valuable and innovative. They need to find a way to the patients. It's definitely a question, my point of view, whether the system will be able to bear a multiplication of the existing facilities and infrastructure that we've invested in for biotech times 3, times 4, times 5, times 10, which might be needed to sustain these products from where we stand right now. And at some point you could say that's going to break the system, and our point of view is, we probably aren't going to see 10 times the stainless steel out there, but what can we do from a technology standpoint to try to use our existing capacity? And that's part of our business model at Wyeth, is to focus on technology. This is just a reflection on the difference between the early days and now. In the early days, we worked on these products like EPO and Factor VIII that were basically replacement factors. There wasn't enough protein in the body. You basically augment that level, and you get much more normal function. These were extremely potent molecules, and so they could be given in very low doses to have their effect. So the technology of the 1980s expression levels was quite satisfactory. But these recent products, many of them are intended to go block a receptor. So you basically need to saturate. You need a lot higher dose for that. And so these are really commodity-type products. And we needed literally hundreds to thousands of fold increase in titer in order for these products to be viable. So we've done a lot of work on cell culture technology over the years. As I mentioned, the huge investment and the difficulty to match capacity to need. And if you've already seen this, it's just a pretty picture, again, just to illustrate the capital investment. This is our Andover facility where Bob Konopaz and I work, about an hour south of here. And this is the Dublin, Ireland Grange Castle facility, as we call it. It's sort of a nice example of a greenfield expansion, if you will. Both of these facilities are about 1 million square feet and represent billions of dollars of investment. Certainly what I'm hearing from our management is they're not salivating to go build another one of these facilities anytime soon. This, for them, was a really big step to go do this. They knew we needed it for Enbrel, which is making right now. But they're not in a rush. What they really want us to do is to see if we can take advantage of these facilities. This is one of the examples that sort of stimulated their interest in our capability to do that. If you just go back and think about how many thousand people, for how long, years and years, it required to build and bring up this facility. This is just a couple people in the lab with a few months of technology development on one particular manufacturing process, where they had invested very little in the early process, but using our basic platform, which I'll speak about in a minute, They had developed a 3 gram per liter process, and then taking some improved process technology that they had been tinkering with in general as technology development activity, and then applying it to this, they got a jump from 3 to 9.6 grams per liter. When you think about building 3 facilities with thousands of people over many years and billions of dollars or something versus a handful of people in the lab, management can see the value of trying to invest in technology, and that's what we're focusing on. The idea is basically to try to limit capital expenditure, so we have this neat little slide. Yes?
Those numbers, is that final product?
Those are our harvest titer numbers rather than final product yield. And as Bob mentioned, the higher we go on titers, and I think I spoke about it earlier, the more of an issue it's going to be for purification, and the more we're going to need to look at more radical technologies downstream to try to accommodate the upstream yield. So it's definitely challenging our sense of inventiveness. And this is just the idea of limiting capital expenditure through technology. And then just to put it in the business context again, this is an interesting slide just looking at the cost of insulin production over the years and the evolution of that. Back in the pre-World War II years, it was very expensive to make, and over many years with process development as well as strain development, the cost went down basically 10,000-fold to become a commodity. So profoundly important gain. And looking at the biotech continuum, we asked the question, where are we here? And we think, the current industry standards that you can expect if you're doing well are a gram or 2 per liter. The liters are a little bit higher than that, and we believe that the future liters are going to be greater than 10 grams per liter out in this kind of time frame. And the way we're doing that is continuing to innovate, to drive down yields and costs. And our mission in Wyeth Biopharma and Wyeth the biotech organization as a whole is to try to make our products accessible around the world. And we think this is really critical if we're going to be able to reach patients around the world, is to drive down costs. This is just an illustration of some technology that I'm going to be showing later, which is using basically expression profiling to try to do improved strain development. And now I'm going to shift into some of the work we've done in different areas, basically on the molecular and cell biology side, to try to address this issue from the cell culture performance standpoint. So we kind of look at this as the building blocks of a cell line and cell culture process, and they're just enumerated here. And I'm going to touch on each of these 4 during the next few minutes, the type of work that we've been doing. Basically, we have found that the choice of the molecule itself actually has an impact on the expression level, so we're looking at that. And then the other building blocks, you start with the protein that you want to make. You design an expression vector with that coding sequence. You're going to end up having to put that together with a host cell. Then you're going to have to make cell lines, which is the cell line development and screening process. And then there's also the use of the development of the manufacturing process with that cell line itself. But I'm going to concentrate on these 4. And we did learn, as I mentioned, that the choice of the molecule is important. In the early days, when we were making replacement factors, the whole goal was to make a protein as close to the natural human protein as possible. So we were making factor VIII. It was really a study of what does factor VIII really look like in the body and how can we mimic that. And it was a giant scientific project over a number of years just to try to make it look as close to natural as possible. We worried about genetic variation in the population for factor VIII and what was the right coding sequence to use and all that sort of thing. Nowadays, many of the molecules never have actually existed in nature that we're making, in which the sequence that we use is being optimized, for example, for binding. And what we found is that during the course of that optimization work, it has a big influence on expression. So rather than just making something, studying it and optimizing it for binding, and then saying time to do process development, well, now let's figure out how it looks. Geez, it turns out this is not easy to express, so I guess we're going to have to build more facilities. We're trying to be more proactive and take that into consideration up front. So what we do is we do a manufacturability assessment basically before we choose the molecule to bring it into development. This is just an illustration of the variation in expression levels. This is in cellular productivity on a per cell basis basically. and shows that there's a big range of expression. Antibodies are known generally to be easier to express than, say, coagulation factors, which are like factor VIII, which is extremely large and difficult to express. But there's also a big range of expression levels within the antibody family itself. Some of the need for doing this, we just don't have the time anymore, and time is truly for us money. We don't have the time to dither like that and find out and go back. We need to figure out fast what we're going to go forward with. We're under a lot of pressure to try to get things into the clinic quickly because, you know, the philosophy is really fail early and often. You just don't know what drugs are going to be worth anything until you get them into patients and find out whether they work. And we used to have, you know, a lot of science that went in, spend a lot of time trying to decide between this and that, and you really can't make the key discriminations until you get into patients. So we've really optimized our process for speed, and we've gone to what we call a platform process development, in which the manufacturing process will look very similar. We used to make custom processes. The manufacturing process will look very similar from one product to the next. That means the same raw materials in manufacturing from process to process, which is a huge savings in terms of time because you already have them all released and ready to go in for GMP manufacturing. And basically, we've standardized the work process so we can make it more predictable and we can move things along. We don't have, you know, the individual saying, I want to try this and I want to try that. That ended up adding time. So we've standardized all of those practices to try to move this along. On the cell culture side of things, that means basically making a cell line and in our lab, going from transfection to the cell line selection in 6 months. And these are just some of the drivers that I've illustrated here. We've got many products that need to get in, speed to clinic, and trying to minimize the investment before what we call clinical proof of concept. So the key thing for us here is that there's just enough time to execute if we're going to do that. There's not enough time to figure stuff out along the way. So we've basically moved the process back, the figuring out, up here to the period just before we begin process development. So essentially, this happens for us right after we've decided what molecule we're going to move forward into the clinic, and that we are going to move it forward into the clinic. We call that the pre-development decision. And as soon as we've chosen that molecule, we just go ahead and make the vectors and crank. So we basically have to figure out with our research colleagues which of the therapeutic candidates we want to go forward with. So the philosophy is essentially for a particular target, the research folks will be focusing on biological activity of the molecule, and we'll focus on the manufacturability of that molecule, and then just choose the one molecule for development. Our role really is on the manufacturability assessment, so I'm just going to speak to that briefly. We want to determine the fit with the platform process development. This isn't an idea of presenting barriers, but really getting that understanding up front. And we want to get an understanding of the unique attributes of the molecule prior to the initiation so there are no surprises. And we do apply this to all the different types of therapies that we look at. My area, since we're cell culture, we focus on expression levels, but we do, as part of the team in the manufacturability assessment, look at biochemical attributes. Aggregate is very important for us, and the ability to remove aggregate, because that's something that with immunogenicity issues that have been seen, we have to focus on for safety reasons, and we need to get that right. So we want to have a head start on that. Product heterogeneity, post-translational modifications, et cetera. And then on the formulation side in particular, the handling characteristics of the molecule in terms of solubility and stability are looked at. And we try to do this on up to 4 candidates prior to the selection of the final candidate, which is quite a bit of work. So focusing on the expression level, we needed a tool that would tell us predictably and from project to project, you know, how does the relative expression of this particular molecule fit in relation to the other molecules that we've looked at. And we had used to use either cost transient expression for that, which is a common way of doing it, or making stable clonal CHO cell lines to consider those to be representative. People were always worried about using pools because they thought the heterogeneity would cause too much instability. what we have settled on actually is using CHO-stable pools for both speed and because they're more representative than any individual clone. If you end up developing a clone, the clonal characteristics may skew your view of where it fits in the overall sort of spectrum of expression levels. This just illustrates that we find that the CHO-stable pool technology is much better in terms of representative than using COS transient. So here we're actually using selection of a whole pool of cells, just a large set of heterogeneous clones in that population, whereas this is just jamming genes in, letting the plasmid replicate in the cells, putting some protein out for a few days, and then you're done. And in this particular case with two antibodies, they both actually were able to force COS cells to make this protein for a few days, but we really found that these two antibodies were very different in terms of their relative expression levels in CHO cells, which is the system that we're intending to use. The predictability of this system is shown here with three different antibodies. So this is doing this CHO pool assessment. It's actually different technology, different vectors, even different host cell, different version of our CHO host cells that's used for that assessment. And it just shows that B versus H versus O as antibodies, this in each case told us that this was going to be a poor expressor, this one sort of middling, and this one a very good expressor. And that's very useful information for us to have up front. This is just some more data showing different antibodies and perhaps also illustrates that even different versions of the same antibody, and this is, if you look at antibody 1, version 1 and version 2, both in terms of the titer and in terms of the productivity that we could gain out of that, there was quite a bit of difference, even with just small differences in the progression of humanization and binding optimization being done within a single antibody, if you will. This is just illustrating that same phenomenon now. I think it's probably the same antibodies here, antibody 1 and antibody 4. You can see that the pool data were showing 1 to be pretty good expressor and quite a bit better than 4. This is the actual data on the clones that were evaluated once we went out and cloned and evaluated these individually, and it confirms that 1 was a better expressor and 4 was not a good one. And this is just an illustration across many projects now where we've done using the same method time and time again. It's very reproducible, the spectrum of different expression levels of the different antibodies that we've created and evaluated. And it's fairly broad and very useful now, because every new project we can start using the language of the best and the worst and go, so where is it? Is it an antibody A or is it an antibody P, if you will? They're just sorted this way. And that's useful for us for when we're starting to plan how many clinical batches, how much tox material we're going to need, how many pilot batches. We have a good handle on it's a good expressor or a bad expressor. And when we're trying to get a lot of projects in, scheduling becomes an important thing for us as well. So this has been really useful for us. And this is just an example of how we use this in collaboration with the research folks, where they were focusing on biological activity, and I just used the two versions of the same antibody, 0.1 and 0.5, that only differed by 4 amino acids. They actually had a 20-fold difference in expression level. And these 4 amino acids were murine to human, the candidates for murine conversion to human residues, and people didn't they wanted to eliminate as many mouse residues as possible from the antibody, and the motivation for that was mostly hypothetical in terms of immunogenicity, and this really won the day, because the difference in expression was so profound that it would have been incredibly problematic to go forward for that hypothetical difference. So that's just an example of how we can use that sort of collaboration and information to really help us to move the project forward. So now moving on, and this is just a little bit more brief than the last section, expression vector design. We used to use what we thought was a very good promoter, and I believe that with that promoter and enhancer system that we were using, we could always get cell lines that reached very high transcript levels, and therefore basically the limitation was no longer how much RNA you had. to synthesize the protein. However, we use gene amplification. This was a DHFR-based system, for those of you who are familiar with that, in which we had to use stepwise selection and methotrexate, and it took months of time, and that's actually a really tough event on the cells in terms of chromosomal stability, which created an additional risk in terms of cell line instability. So we worked on a new version expression vector, and this is what we use now all the time, and it's profoundly affected our ability to do the kind of fast timelines that I mentioned. This is basically now normalized for the number of copies, the levels of transcript, and so the original vector versus the current vector, this vector is profoundly better on a per copy basis, much more efficient in driving mRNA synthesis, and this is an intermediate version that we had along the way. With this, we don't need to do gene amplification. Basically, we just jam genes into the cells, and we put them under high level of selective pressure right away, and some of the cells make enough transcript off of this that even at one or a very few copies, we get enough expression to survive the selection, and those are the ones that we move forward. This is basically a change in the promoter and enhancer that led to that difference. We are also working on other vector improvements besides that which is aimed at the transcription end. This has jumped us so far into the future that we don't feel like we need to invest there that much. We're working on things like the signal peptide and matching the signal peptide to the protein, RNA stability elements on either side of the coding sequence, and things that might enhance the translation of the mRNA as well that go into this. But we're trying to improve this building block as well, and it can make a big difference for us. Another big area for us is what we call host cell line development. So you have the gene now, you have the vector to put it into, it's going to go into a host cell. So this is all within the CHO family for us. And we actually have for many years, I think we published about 10 years ago, used pre-adapted CHO cells because we're going to do stirred tank bioreactors, serum-free suspension. So we pre-adapted these cells for many generations so that they go in very easily. We're continuing to try to develop a better version of those cells all the time as we go along, and then those become the new host for our transfection platform. And one of the areas, we call this like a super CHO cell effort, is trying to study using genomics technology the expression profiles of the cells and find out what makes a good CHO cell. And for this, we've actually turned to the use of both mRNA expression profiling and proteomics. And we're doing this through a collaboration with Dublin City University, which we came to in part because of our development of the Grange Castle facility and relationships and actually employees that had come out of DCU. And we just didn't have enough staff in-house to be able to take on this kind of project. And so we've decided to partner with them on this. We're very busy working on projects all the time, are able to put a little bit of resource on the technology, but not enough for a project of this scope. So the goal of this then is to try to take us to a greater than 10 grams per liter type project. And it's essentially an intellectual partnership as well as a resource partnership in which we each bring important things to the table. We've got many, many years of CHO cell lines and process development experience and an understanding with these, which puts us in a very unique position in terms of trying to do expression profiling and ask the questions, what matters? We also had a substantial infrastructure within Wyeth with Affymetrix gene chip technology, and we decided to leverage that for process development purposes. And we basically had Affy make us a custom CHO chip for this work, and we've applied what we call the first generation chip, which has, I've got it on another slide, several thousand CHO genes sequences on it, and we're in the process of developing a second one that has almost 20,000. I think it's going to be available within weeks if we haven't received it already to this. So what are we going to do with this? Basically, the essential timeline that we use for this type of project is collecting cell samples from bioreactor and shake flask studies of many different types. First you have to decide what you're going to use, then you have to make these through experiments, and then crunch through and do basically make RNAs, do scans and proteomics, and then evaluate all the data. And this is, of course, what everybody says is brutal, and it is, for a period of time. And then essentially what people have said is one opportunity, if you're doing expression profiling, you're looking at things that are over or under expressed, can you actually engineer the cells to constitutively have those genetic characteristics? And the way we're going to do that is basically clone DNAs and use siRNA and gene overexpression to test which of these really work, and then ultimately try to engineer and look at bioreactor performance. This just describes the tools that we use between the different sites. And this was a great deal of work trying to look at the different, what we call industrially relevant phenotypes. What we have are things ranging from cells that grow to high cell density and high viability to high cellular productivity. Obviously, these byproducts are very negative for us, so it would be attractive to have low ammonia and low lactate producers, high growth rates, etc. We collected a lot of samples, a lot of biological replicates for this, and we're in the process now. This is sort of an illustration of what we call the sample matrix. These are all different cell lines and conditions that populate this, and then we do expression profiling and basically make gene chips on all of these. So it's a matter of making RNA, quantifying it, and then doing these scans, and then taking all that data and trying to figure out what pathways are upregulated and downregulated. So we're in the middle of that right now. We've seen a lot of interesting things, and we're in the process of trying to do some of the target identification and validation over the fall of this year. And hopefully we'll have something to share on that early next year that will tell how we're really going to progress on that. And then finally, technologies for cell line development and screening. Then if you're putting the molecule, the vector, and the host together, the process for bringing all these together and to identify great cell lines is something that we've invested a lot in. And with 10 minutes to go, I'm going to try to breeze through this, and forgive me if it's too fast. This is I'm going to skip over this stuff because we've spoken about it. This is kind of an illustration of the speed with which we in more detail, with which we go from the transfection through clone screening and all the way through bioreactor evaluations within a 6-month period, so that we can decide on the master cell bank in a quick fashion. And the key thing to remember here for us is, if we want to find a good cell line, we recognize every transfected cell is unique. We want to sort through these individual clonal cell lines and find the good ones. And in order to do that, we invested in robotics and high throughput technologies to pick and screen hundreds of cell clones. And last night I was looking for a picture of one of these tools. I've got a couple. One of the cooler ones that we've employed, let me just show you this data first. This is just an illustration of what a clone screen looks like for different antibodies. And again, we're seeing, these are individual productivities of individual clonal cell lines being evaluated and then sorted. So obviously the ones we're interested in for any particular antibody are the ones at the higher end. The technology we needed there involved a lot of high throughput, both on the cell culture side and on the analytical side. Because if you remember, we've now grown up little wells of many different clonal cell lines. We got to find out which ones were because they're okay when they're on wells, but if we're going to have to culture them outside of wells, the handwork goes up tremendously, and they're growing. So we need tighter data back very quickly, and we actually have an assay group right within my lab that provides cell culture assay support, and they developed the woman named Judy Chow developed this high throughput tighter assay technology for us, so that we could get an incredibly high number of titer assays performed in a very fast turnaround time, up to 1,500 samples in a single day, results back to people right away. This just illustrates the other cool aspect of this is we now have a high throughput clone picker. We used to do cell cloning by distributing cells into wells of 96 well plates, by limiting dilution and then going through and working up all of these, and we actually had to confirm microscopically that they were clonal on a well-by-well basis, so it was days in a microscope for a couple people for every cloning event. It was very odious, and we hated it. And now this thing goes in, and we plate out colonies, and it goes in and picks them and distributes them into 96 well plates, and you can do all of this in an hour instead. So we do much more cloning now than we used to, because the employees don't mind it nearly as much. These are just a series of other automated technologies that we use, and there's just a couple pictures of the robots that are used in the downstream, the assay side of things to allow us to do these high throughput titer assays. And we can get these. This is our first generation instrument that we used to essentially replace an ELISA. Gave us the readout in 4 hours, and we've now gone to this mesoscale discovery instrument, which does the readouts in 22 minutes. And this has allowed us to have much higher throughput in terms of assays. This is just data showing that the ClonePix works as well as limiting dilution. The results overlaid exactly, basically, when we tested that. And finally, an additional element that we wanted to look at was if we're screening clones, we're aware of product characteristics through that earlier manufacturability assessment that I mentioned. We're aware of product characteristics that might be at issue. Wouldn't it be smart while we're screening clones to also look at those product characteristics? And Judy actually developed a high throughput purification method that could be applied to clones so that we can look at product quality characteristics and use that information in the decisions on which clones to move forward. This is basically more automation, essentially doing the purification in 96 well formats with very good yields and very good reproducibility. And this is an example of applying that to high molecular weight analysis using size exclusion chromatography on 24 clones in this particular case. And so these are percents of high molecular weights, and what we want to do is pick a clone, if we can, that's not way up here in the 8 or 9%, if we can get it down here. This is much less of a challenge for the purification team to be able to purify something if they don't have a lot of aggregate to deal with. So within the project time frame, we can get this kind of data. I think in the interest of time, this last little segment is just about taking cells even further through additional cell line development. And I guess maybe this is just a quick demonstration that when we do it fast like that, we can actually do even better by working on them further. And this is something that we do after we make our phase 1 material, is to further increase the selective pressure on the cell lines. And you can see how we actually got clone distributions that allowed us to get even higher productivity by additional investment in cell line development. It's basically worked for us in many cases with different kinds of molecules where we've gotten significant additional improvements by further working on the cell lines. So in summary then, basically just going back to the theme of innovation, we think it's really important, both in terms of speed and cost reductions, to focus on that. Pipeline throughput is critical for us. IP is still important in this. I guess sad to say, I guess it's fundamental to business, but sometimes it feels very frustrating to have to deal with it. And expanding our output within the capital constraints that we deal with to try to give rise to the cost efficiencies that we need to bring our products to the patients. And I'd just like to acknowledge the folks. These are people who gave me data and materials for this presentation. Essentially all of the stuff that I showed today was presented at conferences within the last year from my lab. So thank you very much for your attention. This is my contact information.
Yeah, hi, thank you. Linda Rafus, Montgomery County Community College. So I know that maybe you said it on one of the earlier slides, but you showed how this worked for antibodies. But did you feel that you are increasing expression levels too for those more difficult molecules like the coagulation factors?
The technologies are much better now, and when we have gone back and used these processes, in particular the screening, we can find cell lines that make more. But the antibodies are still much higher much easier to express. So we're not going to take a coagulation factor from those old expression levels way up onto the right side of those curves, but rather with the full application of all of the vector technology, the cell line evaluation technologies, we usually can find variant cells essentially within populations that are much better. We used to do individual cell line evaluation. So somebody with really working hard might develop 8 cell lines and look at those. And with this kind of power, you can look at 1,000. You're much more likely to find slightly weird ones that are more capable of basically with more ER, more secretory capability. They're able to make more product.
Okay, thank you.
Mm-hmm.
I guess my question is sort of along the same lines. So when you're doing cell line optimization, I mean, there's always what is the gene product that you're expressing in there. So there was always, at least in the academic lab, you might have a cell line that works really great for gene A, but when you try with gene B, it's insoluble or it doesn't produce as much. But you think that by doing this host cell optimization that you'll have like a cadre of cell lines that are superior?
What will probably, we are thinking about it that way so that we might have different versions for different purposes with different characteristics. We haven't really gotten, sort of unique families yet as we had thought about that might be more suitable for one type of molecule than another. We did a little bit of engineering for particularly unique glycoforms that we needed for some of our molecules early on. Mostly what we're doing right now is focusing on performance-oriented characteristics rather than molecule-oriented characteristics because so many of them are antibodies and fusion proteins, and then letting the power of the system actually find, even within that host cell population, the one that works with the genes in combination. And it's quite shocking. My talk at Cell Culture Engineering was about cell line heterogeneity, which I didn't talk about really at all today. But we find a lot of heterogeneity within cell populations. Even if you take a clone, as soon as you start growing it, it's no longer a clone. And we have all kinds of wild things that we've seen that made for an entertaining presentation of some of the challenges we've had trying to maintain essentially stability in the population that's due to clonal heterogeneity within an initially clonal population. But you can also leverage that power to find the good ones that are stable.
Thank you. Locked into the CHOs, is there any developments trying to find alternative cell lines for the production?
We right now are in very little on our side. There are other mammalian hosts out there. CHO is really the dominant one, and largely for us, it's due to the fact that we've invested so much in in-house proprietary media that works well with our cells. And when we've taken even CHO cells from other companies that are partners of ours and tried to work with them, the match between the media and the cells just isn't the same. So that's really a reason not to switch easily to Percy-6 cells is another one that people are working with out there. Baby hamster kidney cells are another one. There are a variety of cell types. But each of them, really, the media development effort is very substantial. And then the process development, we learn how to work with all aspects of our hardware and software and media with the cells. And it's the fit itself that makes it a platform for us overall, which is why we're really focusing 98% of our effort on CHO cell development. We're going to, more than any other reason, we're going to look at some of these other cell lines to see if there are particular advantages of those, either that would cause us to be interested enough to switch, just to be open-minded about it on the one hand, but also to see if those particular characteristics could be looked for in CHO cells as well. Just try to get the best of both worlds.
Just curious, where is the bulk of the IP investment in this process? Is it in media formulations or?
I would say yes. It's mostly in media. Some process, some cell line stuff. When you do find unique things, ways of doing things, we file on that. But mostly it's in the process and the media.
In thinking about the media, how important is it when you're developing these cell lines for manufacturing to get things like serum-free medium and protein-free medium, and what impact does that have on cell long-term viability?
We think it's very important. Basically, from our point of view, we haven't been using serum in any of our in-house developed processes, manufacturing processes, since 1991. We've done all of our work serum-free. A lot of that had to do with the fact that we were developing coagulation factors like factor VIII and factor IX, and the patient population that we were looking to serve were hemophilia patients whose previous products were blood-derived. They were burned big time by blood product-derived HIV and hepatitis, so they were extremely sensitive to this, and they didn't want us putting, bovine serum in our cell culture processes, so we had to get it out early. And there are big benefits to that because you don't have supply issues. Obviously, the BSE issues are much lower. And really, we don't think you need it as long as you know how to work. You have good serum-free media formulations, of which we have one that we're very happy with in-house, but also there are commercial ones that work, depending on the fit with your cells, of course, there. So it's really the way to go. And we actually don't even use hydrolysates in our most of our manufacturing process. All the ones that we've developed in-house, we don't have. They're completely defined media.
What about purified growth factors and
Sorry.
things?
Go ahead.
What about purified growth factors with
That's the only
serum-free?
thing that we put in there is recombinant insulin. But that also can be taken out, and we're right now in the lab, and we did it more than 10 years ago as well. We're developing host cells that are not insulin dependent as well. The last time we did it, it worked fine, but people said the costs just aren't that high relative to the overall manufacturing costs. Don't bother with it. And now when we're trying to squeeze costs more fully, actually it is pretty high. We probably, in clinical manufacturing, I wouldn't be surprised if we spend $1 million or more a year in insulin alone. And when you're getting up, we have 6,000 liter clinical manufacturing facility. At that batch size, insulin is not cheap.
Well, the insulin story is certainly one where the product titer has driven the cost down. You've gotten expression levels on the upstream side. We're not going to get 1 kilogram per liter ever. So what's the cost potential on the downstream side? How far down do you think you can drive costs if you could? How much room is there on the downstream side?
That's a really good question, and we're grappling with that right now in terms of how far should we take this, and really even the shape of the cell culture process will be affected by what the downstream can do. Many of the sort of advanced thinkers in the field think that we need to move back to more traditional, if you want to, more bulk, enzyme production type processes, precipitation type processes, things that are not based on exquisite chromatography, because that's where the facility costs come into play. We talked to all these transgenic tobacco companies and corn companies and all that, and they say, oh, you can do it in the field, you can make the protein for nothing. And we go, okay, so now you make a ton of protein upstream. What are you going to do? You're going to bring it into a facility that looks just like ours, and it's gargantuan, just to make the water and the buffer that you need to be able to purify it, so it doesn't drive the cost down as much as they thought. But I don't know the answer to that question, and we're very worried about that right now. Necessity is the mother of invention, though, so that's a way of looking at it.
So I'm Craig Caldwell from Salt Lake Community College. The research group makes these changes in your clones to increase productivity. I was wondering if those are rational changes or if they do it on a random screening basis.
The way we do it is a collaboration. So we have folks from the development organization that sit with the folks from the research organization. They're doing protein design based on the biological activity, and we are simply, right now, not rationally, just evaluating the outcomes of that. We are starting to do a little bit of modeling to try to figure out whether there's anything in common to that, so that we might actually be able to do design of the molecule for both. But right now, in practice, they're doing rational design for biological activity, and we're just evaluating the outcomes of that and then using that information in the context of their information to make our decisions. But it's a great idea, and we're hoping to be able to figure out, geez, what is there, why do 4 amino acid differences make that much difference in expression? If we could figure that out and start to integrate that into our design process, we may be able to do even better. Thank you.